Hello, everyone, and welcome to today's The Scientist Symposium. I'm Deanna McNeil, Associate Science Editor for The Scientist, and I'll be moderating our discussion. Yesterday, our panelists discussed advances in AAV vector engineering, targeting, and safety. Today, we'll continue exploring the latest research and practical applications of AAV technology in cell and gene therapy. Before we begin, I'd like to thank our symposium sponsors, Vector Biolabs, Shimadzu, Takara, Sinobiological, Beckman Coulter Life Sciences, Biorad, Taconic Biosciences, PHCBI, BPS Bioscience, AdGene, Innotiv, and Mission Bio. We like our symposia to be interactive. We encourage you to send us your questions or comments at any point, and our panel will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsors have provided us with some helpful resources related to today's webinar, and we've posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. And if you've minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. The symposium will be archived on the Scientist website, and we'll send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our first speaker. Nerea Zabaleta is a researcher at the Center for Applied Medical Research, CIMA, at the University of Navarra. She completed her PhD in biomedical research at the University of Navarra and a postdoctoral fellowship at Harvard Medical School. Zabaleta's research focuses on the optimization of AAV-based gene therapy. She investigates different aspects of AAV vectors, including the generation of novel AAV capsids with improved tropism and the optimization of recombinant AAV manufacturing. Let's make sure that our slides are up and running. Excellent, take it away.
Good morning, everyone. My name is Nerea Zabaleta, and I am a researcher at the Gene Therapy for Rare Diseases program at CIMA University of Navarra. Today I'm going to talk about AAV production in a talk titled Less is More, Minimal Helper Genes for AAV Production. This slide contains a clear example of how much difference we can do with AAV in the treatment of several diseases. So here we have a patient that has liver congenital amaurosis and has been treated with an AAV-based product that is called Luxturna. This is a blindness disease, and to evaluate the efficacy of the therapy, the patients are put through this maze test that consists of the patient in semi-darkness following the arrows in the test without bumping into the obstacles in the less time possible. On the left, you can see the patient before treatment that keeps bumping into obstacles, that loses the track, and that needs the help of external people. While on the right, you can see the patient after treatment that is able to complete the test in a very short time. So this is what I call the magic of AAV vectors. These vectors are a simple system composed of a short genome of 4.7 kilobases that has no viral genes or open reading frames, and has only the only viral sequence incorporated in the vector is the inverted terminal repeat, or ITR, which is necessary to encapsulate the genome into the AAV capsid, which is a protein capsid. AAVs have a broad tropism spectrum. There are many natural serotypes that have been isolated, but there are also some synthetic capsids that have been synthesized or modified in the lab. They have the potential to treat many genetic and non-genetic disorders, and this has been shown by the large clinical experience using AAV products, with 8 approved products on the market. However, as you can see here, we have all the products that are approved based on AAV that have different serotypes for different indications, targeting different tissues. However, one of the very clear limitations of AAV is that the price is really high. These doses are $1,000,000 doses. Even though these are one-time treatments, many of the countries and patients cannot afford these treatments. Additionally, as you can see here, the doses that need to be administered are huge. Therefore, as the clinical experience with AAV has advanced, the limitations of these vectors have become very evident. One of them we just talked about, which is the high doses required for therapeutic efficacy. Then there is a specificity issue. These vectors have often off-target transduction of undesired organs, which can lead to toxicity. There is a poor understanding of the manufacturing processes. Immune responses also against the vector have led to toxicities, in some cases to mortality. And AAV has the potential of genotoxicity. We know that AAV can integrate in a very low rate, but still this integration can be risky. We have, in this review that we published a couple of years ago, we have summarized some of these limitations with examples. So therefore, what these limitations do is to lead to lower efficacy, lower safety, and increased cost of AAV therapies. We have several approaches to overcome these limitations. We have two approaches. One of them is improving manufacturing. In this part, we want to describe biological processes behind production in order to understand what drives AAV production and be able to improve it. We also would like to simplify manufacturing to control better the parameters that affect yield and quality of vectors. And we would like to develop broadly applicable manufacturing technologies and methods to be able to standardize AAV production. Our final goal is to achieve better quality and quantity at lower costs. On the other hand, we also work on capsid engineering. So we would like to understand the determinants of capsid tropism, engineer capsids in order to achieve very specific transduction of tissues and cell types. And of course, we want to improve potency and reduce off-target distribution. The final goal is to increase efficiency, safety, and lower toxicity. In this talk, I will mostly focus on the manufacturing piece. AAV manufacturing, there are several methods that are used and have been described. However, the vast majority of people use a method that is commonly known as the triple transfection method. So in this case, and this would be a larger scale process, we have bioreactors that contain packaging cells. Lately, mostly suspension cells are used because it provides several advantages. And these cells are transfected with three plasmids. The first plasmid contains the ITRs and the cassette of interest, which is the recombinant genome that will be packaged into the capsid. A second plasmid contains the rep and cap genes of AAV that are given in trans, and the third plasmid contains the helper genes. So AAV in nature is a helper-dependent virus that needs the co-infection of another virus to complete its life cycle. And this virus can be, for example, adenovirus. So for production, we have a plasmid that contains a big part of the adenoviral genome and has this helper function. So once the cells are transfected, AAVs are generated inside the cells, and there is several downstream processes to purify AAV and obtain a product that is as pure as possible. This process has its advantages. It is easy to set up. Almost every lab that works with AAV is able to produce AAV in-house. It also gives high yields at research grade scale, which it's very convenient for early development in early proof of concept therapeutic studies, for example. It has a short production timeline and it is adaptable to suspension cells. However, there are several disadvantages, such as the lack of scalability, the high costs on plasmid production and transfection, and also it is complex to scale up the transfection. There are also reproducibility issues, which is an issue for market products or clinical products. And finally, a big important issue is the impurities. So the AAV products that we produce have several impurities. For example, empty capsids, partially filled capsids, contaminants of, for example, DNA contaminants from the plasmid backbones or the packaging cell genome, and all these contaminants and impurities can lead to toxicities. Therefore, in this scenario, our main goal is to engineer the transfected elements and the packaging cell line to optimize AAV yield. So, given the current production method, we can make improvement at different levels. Here we have focused on the upstream levels, specifically on the transfected elements. And today I'm going to talk about the work of a very talented postdoc called Jiten Doshi. So when we started to tackle this issue of the transfected plasmids, we realized that there was an issue with the helper plasmid. So we went back in literature and we saw that since already the 19th, it had been described that there were three main helper genes that came from adenovirus, which are E4, ORF6, E2A, and VARNA1, that were required for AAV production. And this was pretty well established. However, when we went to check the helper plasmid that was used, so for example, in this case, this is delta F6, which is a plasmid that it is used in many labs to produce AAV as a helper gene, we saw that there are many open reading frames and many genes of adenovirus that are included. So we have a piece of 12.5 kilobases of the adenoviral genome in this helper plasmid. This is a very big plasmid and a very big sequence, and considering the cell genes that were described, we had way too many genes included there and way too many sequences that were a risk in terms of toxicity, for example. So to do our experiments, we work with the different plasmids. So we transfect this plasmid in cells. So we have our AAV genome, which is a CMV luciferase. We have a rep cap plasmid that has the rep of AAV2 and cap of AAV9. So we would be producing recombinant AAV9 vectors. And we have several different plasmids that contain the helper genes. So for example, delta F6, and we also have plasmids that have the individual genes. We transfect these plasmids in equimolar ratios into HEC293 cells in 24-hour plates, and 3 days later, once AAV is produced, we do 3 freeze cycles to lyse the cells and release AAV. And with the supernatant, we run a qPCR to test the vector titers, and we also ran transduction assays in HEC293 cells to analyze the luciferase expression. So in our first experiment, we just wanted to test if the transfection of these 3 genes alone was able to produce AAV instead of transfecting delta F6. So you can see that when we used delta F6 versus these 3 genes, we were obtaining around 10-fold lower vector titers, which means that these 3 genes are not enough for AAV production, and that somewhere in here we have another helper gene. So what we did first was to eliminate these parts of genes that, because of their function, because they are, for example, structural genes, we anticipated that were not needed for AAV manufacturing. So once we removed these parts, we have two different versions of the vector, of the helper plasmid, we see that the titers of AAV are not affected. Therefore, we determined that these genes are not essential for AAV production. Then we focused on the E4 gene. So the E4 gene has several open reading frames, and what has been described is that open reading frame 6 is the one that is essential. So to test if there was another open reading frame that was required for AAV manufacturing, we generated different versions of this gene. So we have the full gene controlled under a CMV promoter. We have the E4 RF6 control under a CMV promoter. This is the essential gene previously described. We have the full gene with the endogenous promoter, and we also have the open reading frames 6, 7, which is two open reading frames that partially overlap. When we produce vector using this helper, we can see that with delta F6 we have the expected titers. When we transfect the three pre-described helpers, we have, as expected, lower titers. When we use open reading frame 6, 7, we don't recover the titers, and we use the full E4 gene, we don't recover the titers either. So the helper essential gene that we are looking for, it's not in E4. Then we focus on the DBP gene, or the DNA binding protein gene, that has several open reading frames, among them the E2A, which is an essential gene, essential helper gene, and we generated this version, so plasmid containing the 2233K, which are two open reading frames that also overlap partially, and another one containing 100K. When we test this plasmids, so we add the three essential genes and or 100K or 2233K, we see that when adding 2233K, we are able to restore the AAV production titers to what we expect when using delta F6. Therefore, with this first set of experiments, what we have seen is that with the pre-described essential genes, we have a lower AAV production. However, when we add 2233K on top of it, we achieve levels of AAV production that are in line with delta F6, or with what we would expect from a regular AAV production. However, this made us question if all of these 4 genes are required for AAV production, or if all of them are essential. Therefore, what we did next was to combine these 4 helper gene plasmids in different combinations. So first we have our control, which is delta F6, where we obtain the AAV production levels that are expected. When we just use a single helper gene, we do not have any AAV production, or it is close to background. When we combine them 2 by 2, we start to see some production happening in some of the cases. And interestingly, we see that when we use E2A and 2233K together, we have decent production levels, which are more or less 3-fold lower than what we achieve with delta F6. When we combine them 3 by 3, we see that we achieve very good levels with E2A, VARNA, and 2233K, which are in line with what we achieve when we use all 4 genes together, and in line with delta F6. Therefore, what we conclude from this study is that the minimal helper gene set is 22K, VARNA1, and 2233K. And contrary to what was believed until today, E4ORF6 gene is not essential, and it doesn't even provide an improvement in AAV production. We wanted to go further and see if 22K or 33K were doing something, and if we could use only one of the open reading frames. So we divided these ORFs in 2 different plasmids, and we tested them independently. You can see that when we have only 22K, we have a decent production, but it is significantly lower than what the titers we have when we use both of them. And when we only have 33K, the production levels are much lower. So from here we have concluded that 22K is probably the most important open reading frame for AAV production, but 33K is likely also having an effect and providing some advantage on AAV production titers. Okay, so once we have defined the minimal helper gene set, we wanted to rethink the helper plasmid. So the plasmid that we are using right now is a very big plasmid. So it contains 12.5 kilobases of the adeno genome, and we have created two versions of new helpers. So one of them contains the 4 genes, also E4ORF6, because this is the gene that was previously described to be essential. And then we have another plasmid that contains only 2233K, E2A, and PARNA1. This new helper is shorter in size by 7 kilobases. So this gets us to much lower DNA amounts that we need to transfect and lower cost of transfections, of course. So when we test these versions, and here we have also included another helper plasmid, which is ALDX80, which is a helper plasmid that also contains a part of the adenogenome, which is also very large. We see that with our new versions, we obtain titers that are in line with what we obtain with the traditional helper genes. We have also tested the potency of these vectors, and here we have transduced HEC293 cells at the same MOI levels with the vectors produced with the different plasmids, and we see that the potency of the vectors remains the same independently of the helper gene that we use. From that study, we also show that we confirmed that E4ORF6 it's not necessary. So we made our scale-up studies only with the helper plasmid that contains 3 genes. And here we have produced different batches of AAV at a 500 milliliter scale, which is a scale that we use in the lab regularly for example, in vivo studies. And we see that the production levels are in line with what we obtain with the traditional helper gene. Additionally, as I told you before, for these studies we have used AAV9, the capsid of AAV9 to produce recombinant AAV9 vectors, but we have also looked at different serotypes and different genomes as well, and we see that our observations remain. So we have good levels of production with equal vector potency independently of the helper gene that we use here. So once we got here, we wondered, so we have created the plasmid, which is a great tool, and it's something that actually is available in a gene for anyone that would like to use it, and we think that it significantly improves the AAV production process, reduces the cost, and reduces the potential for impurities. But we wanted to go a step further, and we wanted to create a helper cell line. So we wanted to integrate these helper genes in the HEC293 cells in order to be able to have a line that doesn't need the transfection of this helper gene. So at first we tested the stable integration of all the 3 genes. However, the VARNA1 gene is a non-coding RNA, and we have no way of controlling the expression of this VARNA, so we cannot have an inducible system, for example, to express the gene, and therefore these cells were not stable enough, and with time they lost the capacity of producing AAV. So while we are looking for solutions to integrate VARNA in a controlled manner, we have created cell lines that have integrated the E2A and the 2233K genes. So we have used the piggyback system, and we have this construct that has a bidirectional TRE promoter that controls E2A and 2233K, the expression of these two genes. And we also have, under a different inducible system, we have a fluorescent protein in order to sort the cells. So we transfect this plasmid together with the piggyback plasmid, and after random integration, we induce the fluorescent protein, and we have cells that become green. So we sort these cells, and we have created polyclonal cells, but also monoclonal cells. And we have studied these monoclonal cells. So first of all, does the inducible system work? As I said, we have a dual bidirectional TRA promoter, and when we test this construct in plasmid format, we see that this is the plasmid. We see that when we do not add any induction, we don't have production. But when we add the inductor, we obtain high levels of production in line with what we obtain with our control delta F6. So when we go to the cells, what we see is that we have, and you cannot see this because I think you are seeing my image, but with our system, when we don't add the inducer, we don't have production, which is good because it means that there is not leakiness. When we add the inducer plus the VARNA, we have very good levels of production that are in line with our control, and this is the triple transfection control. So these are not stable lines, stable cells. But when we add only the inductor but not the VARNA, so this would only have as helper E2A and 2233K, we still have production, which is like 2 to 3-fold lower than what we expect with a regular delta F6 production, which we had seen before. Importantly, this production is stable over time, so this is 70 days post sorting the cells, and we see that the production levels are remain stable during the time until reaching this point. In conclusion, we have identified the minimal set of helper genes. We have identified 22K, 33K as essential genes, which weren't described before, and we have determined that E4 or F6 is not an essential helper gene. We have generated a new helper plasmid that is 7 kilobase shorter, that has the same production efficiency. And finally, we have generated a stable cell line that eliminates the need of the transfection of helper genes. Finally, I would like to thank everyone in the lab, especially Jiten Doshi and Yuning, who has worked in this project, my colleague Anil Chekuri, and my mentor Luke Vandenberg, and of course the funding bodies, with a special mention to ACUOS and MLSC. Thank you so much for your attention. I will be very happy to take any questions.
Fantastic. Thank you so much for that wonderful presentation, Nuria. Let's jump into our Q&A. How many copies of the helper genes need to be integrated to achieve good production levels?
Yeah, so yeah, that's a great question. By the way, before I start answering, I wanted to make sure, because I see that the video at the beginning wasn't working. So you can find the video in the handouts, and you can see it. It's publicly available in the Luxturna website as well. So you can go and see it. It's a very nice video. So regarding the copies, we have seen that in our clones we have between 10 and 20 copies, and those are the clones that work best. So yeah, I think around that 10, 20 copies we would be fine. With less copies, we don't see as good production in the clones.
Perfect, thank you. And thanks for mentioning the video as well. Have you defined the optimal amount of expression needed for each helper gene?
Yeah, so yeah, we have done several studies, and we have, as we have the helper genes separated in different plasmids, we have worked with, maintaining two of them with a fixed amount and then increasing or decreasing the other one, looking at mostly molarity. So, like twofold higher, one, half, and everything in between. And we don't really see that much of a difference with any of the three helper genes that we have identified in the study. So we believe that, once you go up very high, you start seeing lower titers, and probably that's because these genes are toxic if they are expressed in a very high amount. But yeah, we I think the equimolar ratio is probably the best at this point, or that's what we have determined.
Excellent. Have you considered performing a targeted integration of these genes?
Yeah, that's a really good point. We have considered it. We haven't done it yet, to be honest. As I said, we need a fair number of copies of the genes, and we are using promoters that are pretty strong. So we would need to carefully design it in order to have integration in a place maybe of several copies. But that would be a much more controlled way of establishing the stable cell line. So yeah, that's a great idea.
Excellent, thank you. You mentioned VA-RNAi, some trouble with those cell lines towards the end of your presentation. Have you generated a stable cell line with VA-RNAi?
We have not, and we are exploring several strategies, um, which some of them seem to be successful in a way. We don't achieve the full production level that we achieve with VA-RNA, but we have some alternatives. It is also true that we think that as I showed with E2A and 2233K alone, we already have a relatively good production, which is around threefold lower than what we see with when we include VA-RNA. So we are wondering if in the future we have fully stable cell lines where we have only the two helper genes, if that's going to give us already an advantage, just the fact that all the cells have all the elements to produce AAV. And maybe we do not need, or VARNA is not as essential, because at this point I believe it's more an enhancer than an essential gene. So we are still exploring it, and I think ideally we would find a solution. But if we don't, I think in a stable cell line context, we still might be fine.
I see, thank you. Maybe one last question. Could you tell us what is the most exciting avenue for your research, in your opinion? What are you looking forward to in the future?
Um, yeah, that's a good question. I think for us, the engineering part is important, and it is very relevant. But I think the biology, it's probably the most important part for us, because understanding the biology behind manufacturing is going to probably give us the key to unlock some of the issues that we have with AAV manufacturing. So I think the most exciting would be to, for example, find host determinants of a good production. So to see if the genes in the HEC293 cells, in the producer cells, uh, have an effect, which genes, and if we can modulate them in a way by engineering the cell line or using small molecules. And of course, looking at the elements that we use, the ones that we transfect as well, and see what is the biology behind them. For example, what is the role of 2233K in AAV production. I think those, if we answer those biological questions, we might be in a much better position to really improve production to, like in a significant magnitude, probably one order of magnitude higher.
Perfect. We'll keep an eye out for that. Thank you so much, Nerea. That was a wonderful presentation and a great discussion.
Thanks so much.
And now let me introduce our next speaker. Hirotaka Kuroda is a researcher and developer in the Cell Analysis Department at Shimadzu Corporation, a leading Japanese company in science and technologies and manufacturer of analytical and measuring instruments. He embarked on a career dedicated to advancing culture media analysis techniques and their applications in the biopharmaceutical field. His current passion lies in streamlining culture process development as he pursues a PhD in engineering at Osaka University, focusing on culture medium design methodologies that promise to enhance bioprocessing efficiency. Let's make sure that our slides are up and running. Perfect. Take it away.
Good afternoon, everyone. My name is Hirotaka Kuroda, and I work in Cell Analysis Department at Shimadzu Corporation in Japan. I am very pleased to have this opportunity to speak today. In my presentation, I will mainly introduce the history of medium development and also highlight some of the latest technologies. I hope this presentation will be an opportunity for you to reconsider the culture media you are currently using. And since many of you may not yet be familiar with my company, Shimadzu, so let me introduce my company at the end of this presentation. So, I will begin with the history of medium development. Now, I would like to provide an overview of the history of culture medium development for mammalian cells. As the title of this presentation is Culture Media Development 5.0, I classify the history of development into 5 phases. In phase 1.0, around 1900, researchers first succeeded in vitro culture using mammalian cells. Then, up until around 1950, studies advanced to identify what components are important for cell culture. Next, the development of serum-free media began, and from the 1970s onward, with the progress of analytical instruments, researchers began analyzing both the culture media and supernatant. Finally, in the current phase, the major trend is efficient design and optimization of culture media based on the mathematical models to develop the customized media individually. In the lower left, I show the trend in the number of publications related to cell culture media. As you can see, in recent years, several thousand papers have been published every year. Through today's webinar, I hope you will understand that much research supports the culture media we are currently using. I would also be very pleased if you could connect the changes in this graph to the 5 phases, and at the same time get a better understanding of the latest method in culture media development that you can apply today. Now, I'd like to move on to the individual phases, starting with phase 1.0. The earliest example of maintaining animal tissue outside the body was achieved by Linger. He developed a Linger solution consisting of inorganic salts. Using this solution, he achieved keeping a frog's heart beating ex vivo. Based on Linger's work, many researchers attempted to use inorganic salt solution not only to maintain tissues, but also to cultivate cells. However, the cells did not survive in inorganic salt solutions. As a breakthrough, Ross Glanville-Harrison reported that frog nerve fibers could grow when cultivated in lymph fluid. However, later studies reported that lymph fluid was not suitable for mammalian cells. Alexis Carrel and his colleagues discovered that blood plasma was more appropriate for mammalian cells, and blood plasma then became a major culture medium. Based on these studies, during the phase 1.0, the trend was to cultivate mammalian cells using body fluids. At the same time, researchers became interested in identifying which specific components within these body fluids were essential for cell culture. Therefore, in phase 2.0, many studies were conducted to identify the key components required for cell culture. Two main approaches were taken to achieve this goal. The first was the reductive approach and the second was the synthetic approach. In the reductive approach, researchers gradually narrowed down the key components from body fluids step by step. A well-known example of this approach was the work carried out by Alexis Carrel and Lillian Baker. They purified the protein under mild conditions without denaturation and successfully calibrated fibroblasts using its solution. Furthermore, they discovered that partial hydrolysis of proteins exhibited even higher growth-promoting activity. Through this, they identified that proteins, and especially their hydrolysis within embryonic extract, is important for cell culture. However, the reductive approach had limitations to identify important components at the detailed molecular level. Therefore, the synthetic approach was developed to advance molecular level understanding. Representative studies the work of Albert Fischer and Harry Eagle. They filtered serum using dialysis membranes to remove only the low molecular weight components. They found that in the dialyzed serum fraction, which contained only macromolecules, cells could not grow. So they tested one component after another to see what needed to be added back to enable cell culture. As a result, in 1959, Eagle developed the minimum essential medium, MEM, which is still used today. MEM consists of glucose, 6 salts, 13 amino acids, 8 vitamins, and dialyzed serum. Next, in phase 3.0, researchers began developing serum-free media. The use of serum presented several drawbacks, such as lot-to-lot variation and the risk of viral contamination. Raymond Parker developed M199 composed of more than 60 components, and they successfully cultured chicken embryonic cells. Later, in 1965, Richard Ham developed Ham's F12 to culture CHO cells. In previous version Ham's F10, serum had been replaced with albumin and fetching, but in F12, these proteins were successfully substituted with linoleic acid and ptolysin. One important insight was that serum-free media are not universally applicable to all cell lines. Instead, serum-free media are cell line specific and require customized optimization. Serum-free media were further accelerated by the rise of biopharmaceuticals. In 1986, the world's first therapeutic recombinant protein tissue type plasminogen activator was approved. Since then, many biopharmaceuticals have been manufactured by cell culture, leading to rapidly increasing demand for GMP-compliant serum-free media. Phase 4.0, beginning after 1970, was characterized by the trend of analyzing culture supernatants. Because cells consume and metabolize the components in culture media, the medium environment is constantly changing. Therefore, it is important to observe these changes and design balanced media where nutrients do not become depleted and toxic metabolites do not accumulate excessively. This new focus was supported by the progress in analytical technologies at the time. After 1950, analytical technologies such as chromatography, NMR, and mass spectrometry were developed. Researchers then applied these technologies to the analysis of media and culture supernatants. However, many analytical technologies have been developed. The analysis of culture media and supernatant was complicated and limited throughput. Because the chemical properties of medium components vary widely, separate analytical methods had to be built for each category, such as amino acids or vitamins. In addition, culture media contain both highly concentrated and trace components. Therefore, in order to measure all of the components simultaneously, researchers had to prepare multiple dilution series of analytical samples. To address these challenges, we developed the LCMS method named cell culture profiling, which is optimized for the analysis of culture media and supernatant, launched in 2015. Since then, we have updated the method twice, and it is now available as version 3. With this method, up to 144 components in culture media or supernatants can be analyzed simultaneously in less than 20 minutes. This method is ready to use and can be applied immediately once installed on Shimazu LC-MS system. In addition, the sensitivity has been carefully adjusted so that both high concentration and low concentration components can be analyzed with only a single dilution series. For highly concentrated components, we intentionally lower the sensitivity to avoid signal saturation. For trace components, on the other hand, we maximize the sensitivity. As a result, even trace components can be detected with good peak shape quantitatively like this. On this slide, I will present two application examples of our comprehensive profiling method. The first case study is batch culture of antibody-producing cells. The culture supernatants were sampled over time and analyzed, with 74 components detected out of 144 registered. This time-course visualization shows how certain components were depleted during the culture and how certain components exhibited metabolic shift, such as the lactate switch. Such monitoring can help improve culture performance by supplementing or adjusting these components. The second case study demonstrates how adjusting feed composition improved antibody quality. The researchers noticed sequence variants during antibody quality analysis. Therefore, using Shimazu profiling method, they discovered that proline was depleted during the culture, as shown in this blue graph. By increasing the concentration of proline in the feed medium, they successfully prevented depletion. As a result, quality analysis confirms that the desired native sequence antibody was obtained. These examples highlight that time-course analysis of multiple components can provide key insights to improve culture performance. Now we move on to phase 5, which is current trends. In recent years, because serum-free and chemically defined media are required, optimization of culture medium compositions tailored to specific purposes has become increasingly important. The number of related publications has also been rising sharply. However, the possible combinations of medium components are astronomically large. Because cell culture experiments are both labor-intensive and costly, testing all combinations is simply impossible. Against this background, there has been growing interest in using mathematical models to achieve more efficient and precise medium optimization. Approaches based on mathematical modeling can be broadly categorized into two types: mechanistic models and data-driven models. Mechanistic models are based on existing biological knowledge, therefore offering high interpretability as well as robustness and transferability. On the other hand, data-driven models can capture complex nonlinear interactions among medium components. I will begin by introducing mechanistic models, focusing on two representative methods: genome scale modeling and kinetic modeling. On this slide, I will explain the concept of genome scale modeling. Genome scale models describe all stoichiometry-based metabolic reactions. They allow us to estimate the fluxes of individual metabolic pathways combined with flux balance analysis (FBA). The process begins with collecting the full genome sequences of the target species from experiments or databases, and then all metabolic reactions are listed. This draft model is then curated by adjusting reaction directions and adding pathways such as circulation process that cannot be described by genome sequences alone. The result is a generic model. Next, cell line specific data obtained by culture experiments and measuring parameters such as specific consumption rates of medium components. Incorporating this data allows the construction of a cell line specific model. The final set of metabolic reactions and the flux B can be represented in the matrix format. Then subsequent FBA assumes the steady state and estimates the fluxes of all cellular metabolic reactions. When conducting FBA, experimentally measured data such as specific consumption rates that are obtained when constructing specific models can improve the accuracy of flux estimation. This full process using genome-scale models enables us to identify bottlenecks in the production of targets and to simulate the effects of medium composition by changing specific consumption rates. Many of these models are already published in the papers. For example, ICHO models are commonly used for CHO cells, whereas Human 1, a genome-scale model of human metabolism, is used for HEC cells. Therefore, when using genome-scale models, researchers can refer to these existing models. On this slide, I will introduce the studies that apply the genome-scale model to identify metabolic bottlenecks and to simulate optimal medium compositions. In this research, CHO cells were cultured in fed batch mode using two types of basal media and two types of feed media, resulting in 4 different medium conditions. The result shows that when feed medium A was used, the titer was higher compared to condition with feed medium B. Furthermore, in the conditions with feed medium A, the specific consumption rate of asparagine and aspartate were higher than feed medium B. This experimental data was then used as input for genome-scale model to estimate intercellular fluxes. Among the 4 conditions, the first and the third boxes in this diagram represent the cases using feed medium A. The results suggested that the fluxes of asparagine, aspartate, and their entry into TCA cycle were higher under feed medium A than under feed medium B. Based on these findings, the composition of asparagine and aspartate in the medium was considered to be important. Therefore, by simulating different concentrations and ratios of these components using the genome-scale model, it was demonstrated that their balance has a significant impact on culture performance. This indicates how genome-scale modeling can provide valuable insights for medium optimization. Now, let me move on to kinetic modeling. Kinetic models are dynamic mechanistic models that estimate kinetic parameters from data to reproduce and estimate culture behavior. For example, in batch culture, the change in viable cell concentration over time can be expressed by this equation. Here, mu represents the specific growth rate and Kd represents the specific death rate. The specific growth rate mu is often described using the well-known mono equation. Similarly, the time course changes in medium component concentrations can be formulated using their specific consumption rates. In this way, the culture system is mathematically described by integrating knowledge-based equations. Then, parameters in equations are determined using experimental data or values reported in published papers. With these parameters, computer simulations can be performed to estimate culture performance and medium component trends. A key feature of kinetic modeling is its ability to describe the time course changes of each component concentration during culture. Currently, applications of kinetic modeling are actively being applied. As an example, here I show a study that kinetic modeling was applied to AAV production. Next, I'd like to introduce data-driven approaches, which represent another important direction in medium optimization. In data-driven approaches, statistical and machine learning methods are employed to directly model patterns from datasets. In recent years, the iterative strategy called sequential optimization has gained attention. Here is a conceptual diagram of this approach. First, initial experimental conditions are designed, often using DOE methods. For example, in this case, we assume a scenario where three medium components are to be optimized. Next, cell culture experiments are conducted according to the experimental plan and performance data of interest are collected. If the target performance is reached, medium optimization is complete. Otherwise, the new data are fed into the algorithm, the model is updated, and new conditions are suggested. By repeating this cycle, the optimal medium composition can be efficiently identified. This iterative strategy is now becoming mainstream in data-driven approaches. With this background, we have also been developing culture medium optimization technologies using data-driven approach. We are collaborating with the Japanese AI startup Epistolar to develop a technology called CellTune, which applies AI to optimize cell culture conditions. CellTune is a software platform that enables users to perform the sequential optimization strategy I introduced on the previous slide. You may wonder whether CellTune is just another AI tool. The answer is no. CellTune achieves higher efficiency than conventional AI by employing its own proprietary algorithms. So on this slide, I will give a brief explanation of the algorithm in CellTune and then show some examples of its performance on medium optimization. CellTune employs Bayesian optimization, which enables the sequential optimization I explained in the previous slides. Additionally, proprietary Epistolar's algorithm incorporates insights from the life science field. For example, they emphasize robust conditions that are less influenced by life science experimental error. As a result, demonstration data from two types of optimization scenarios shows that our AI can increase the percentage of runs achieving the target performance compared to standard Bayesian optimization methods. Furthermore, our AI achieves optimal outcomes with fewer experimental iterations. Finally, on this slide, I'd like to introduce a case study that CellTune was applied to optimize a feed medium. In this study, the concentrations of certain components in feed medium were optimized to improve antibody production. The optimization experiments consisted of 3 cell culture rounds with 12 conditions tested in each round. As a result, compared with the highest value before optimization, the antibody titer improved by approximately 40%. This history plot shows the titers of the 12 conditions in each round. As you can see, in the third round, the overall antibody titers were consistently higher. This summary plot compares the concentrations of the 13 components across the 12 conditions in round 1 and round 3. In round 1, component concentrations varied widely, but in round 3, they showed much less variance, suggesting convergence toward optimal values. Like this, CellTune also provides rich visualization features, allowing users to clearly observe and interpret the optimization results. Now, let me summarize. Culture medium development began in the early 1900s with cell culture using body fluids. Researchers then worked to identify the critical components of these fluids, leading to media such as MEM in 1959 and HAMS-F12 in 1965. These breakthroughs sparked rapid growth in medium-related research. After approval of the first therapeutic protein in 1986, research accelerated further, also supported by advances in analytical technologies. Since 2010, efforts have focused on medium optimization for specific purposes. I hope my presentation has given you a clear overview of the history, current trends, and technologies in medium development. At the end of my presentation, let me briefly introduce my company, Shimazu, in the next 3 slides. This year makes Shimazu's 150th anniversary a memorable milestone. Our business consists of 4 main segments. Over the past 150 years, we have cultivated fundamental technologies that allow researchers to observe what is invisible and perform quantitative analysis. Our cell analysis business originated from the analytical and measuring segment. We have advanced various technologies such as culture media analysis, microscope image analysis, and the automation of cell culture procedures. As today's presentation has focused on culture media, I'd like to highlight our business in cell culture media. As I mentioned earlier, we have developed dedicated LC-MS methods to analyze culture media and supernatants, as well as AI-powered software for medium optimization. In addition, Shimazu Group's long experience in medium development enables us to provide custom media with optimized formulations. Through these technologies, we support you to improve the efficiency of bioprocess development. I hope that you are interested in our culture optimization technologies. The best way to understand their value is to experience them firsthand. That's why we offer trial services. Specifically, we provide 2 types of trials: LC-MS analysis of culture media and medium optimization using CellTune. Since the number of trial opportunities is limited, if you are interested, please feel free to contact us for more details. Thank you for your attention today.
Excellent. Thank you so much, Hirotaka. Let's jump into our Q&A. Can the optimization algorithm developed for CellTune also be used to optimize factors other than medium components? I'm having a hard time hearing you, Hirotaka.
Okay. Okay.
Oh,
Hello.
hello. Thank you so much.
Okay, so could you repeat the first question again?
Yeah, absolutely. Can the optimization algorithm developed for CellTune also be used to optimize factors other than medium components?
Okay, so thank you for the question. The answer is yes. Actually, Celltune is not limited to medium composition. Celltune's core proprietary algorithm can be applied to various types of parameters. So for example, we have successfully applied the algorithm to culture-related factors like pH and even practical parameters such as pipetting steps. And in context of AAV production, it can be used to optimize key parameters such as plasmid ratios or transfection timing. Yes, even though today's my presentation focus on media development, but Surgeon can flexibly optimize those media parameters too. So you can set any parameter you are interested in.
Perfect. Thank you. As we know, there's variability in cell culture, and there can be day-to-day differences even when conditions are the same. What considerations should be taken into account in the medium optimization process to address such variability?
I see. I see. Thank you for the excellent question. Let me share two practical points based on my research experiences. First, always include a control condition with at least 3 biological replicates. This is a very basic point for the researchers, but this helps you see whether variability comes from day-to-day shift or from same day, same condition noise. Yeah, so you can distinguish that. If the issue is from day to day, compare the results after normalizing by that day's control. You can compare the different days samples. And second is, so please be sensitive to small changes in the medium. For example, store media properly, which means protect from light. And for example, when you add some components to the media, please confirm the final pH. This may sound very simple, but in practice, they can have big impact. So this awareness reduces variation and catches major mistakes early. Also, I recommend you to talk with your team about little details in daily experiments. So other members often notice what we miss. So yeah, this is my answer. I hope that answers your question.
It does. Thank you. Um, what kind of progress do you anticipate in the next era of culture medium development? So, for example, looking forward 10 to 20 years from now.
Okay, I see. Thank you for the big picture question. This is like medium development 6.0. I like to highlight two directions. The first is hybrid modeling. Recent work is very hot on interesting data-driven approaches and so integrating, so very hot on integrating data-driven approaches and mechanistic models. The motivation is to offset the weaknesses each other. For example, machine learning can capture the complex changes, but it has limitations in generalizabilities. So combining mechanistic models can provide interpretability, robustness, and so on. So hybrid modeling is one of the next area. And the second is about the automation, the convergence of AI and the laboratory robotics will create closed-loop platforms that can conduct design experiments, execute them, and learn from the data and immediately provide the next conditions. Actually, Shimazu Corporation is pursuing this automation system under the new concept called autonomous lab to establish this system. Okay, so thank you for your question. That's all.
Excellent. Thank you. The black box nature of AI could become a factor causing hesitation in its on site use. What are your thoughts on that?
Okay, so this is also a very good question. Yeah, so I'd like to introduce two approaches. The first one is to incorporate mechanistic models, as I mentioned. Yeah, so if the mechanistic model alone predicts well, it is very useful for your work. And yes, also, as I mentioned, hybrid modeling is also one of the candidates. And the second is AI, but explainable AI. So not all AI is a total black box. Actually, some AI methods let us understand why the decision was made. Yeah, addition to this, I recommend you to visualize your result by yourself and reanalysis to check which variables are important. Actually, Searchon, as I introduced, Searchon can output graphs that show which variables are important for the prediction. So it helps you to interpret the results. So I hope you can apply Searchon technology to your work. Thank you.
Excellent, thank you. I think we have time for one more. Regarding the Zeltune software, since it requires uploading data for use, what measures have been taken to ensure its security?
Yeah, nice question. Yeah, so security is very important for us too. Searchon current version works on an internal server PC installed at the customer's site. So there is no need to expose that server to the external internet, which greatly reduces leakage risk. And we also plan a cloud version of the Searchon currently. And in that version, so we're designing it with security as a top priority. Yeah, so as you mentioned, security is very important. So we focus on that. Thank you.
Fantastic. Thank you so much for that presentation and that conversation, Hirotaka.
Thank you so much.
We'll now take a brief 5-minute break, and we'll be back with our next presentation at 11:17 Eastern Time. See you soon. Hello, everyone, and welcome back. I'm happy to introduce our next speaker, Matthew Hirsch. Hirsch earned his PhD in bacterial genetics from West Virginia University and completed a postdoctoral fellowship in investigating AAV under the mentorship of Dr. Jude Samulski. Now an associate professor in ophthalmology and microbiology at the University of North Carolina, his research focuses on elucidating AAV biology towards enhanced AAV gene therapy for ocular disorders. Hirsch has co-authored more than 50 peer-reviewed publications, founded several UNC-affiliated biotech companies, received multiple U.S. patents, and has been honored with the American Society of Gene and Cell Therapy's Outstanding New Investigator Award for his contributions to the field of AAV gene therapy. Importantly, Hirsch and collaborators have multiple programs that are approaching clinical applications to improve vision loss for genetic and acquired ocular diseases. Let's make sure that your slides are up and running. Perfect. Take it away.
Hello, folks. It's Matt Hirsch from Chapel Hill. I'd like to thank the scientists for inviting me to give a talk on some of our work. This is kind of a maybe 5 to 10 year learning experience where we've encountered some things on our way to trying to help a particular rare disease. And the title of my presentation today is From All for One Towards One for All. Augmentation of an ocular immune privilege for the treatment of multiple diseases via gene therapy. I'm currently located in Chapel Thrill, and it's a beautiful day here in North Carolina. I'm a member of the old Gene Therapy Center, which is now the Center for Molecular Medicine, and part of the ophthalmology department as well. Our lab in general, just as a quick intro, focuses on preclinical drug development, and we worked on the muscle, the liver, and in the past 5 or 10 years or past decade, we've been working in the eye. And we also work on the biology of AAV vectors and study the inverted terminal repeats to alter the cellular host response upon AAV vector transduction towards increased transduction efficiency and safer gene therapy. But this will be a preclinical program talk, and I have the outline right here. Quick background, and then we'll talk about the first couple years that we spent on this project doing an all-for-one approach, and this is AAV gene addition for a rare ocular disease. I think we got to a point, we wanted to see it move forward, and it was just a little dose of reality where we hit a roadblock. And then, after running into the stone blocking the road for about 2 years, we decided to go around it. And that path actually took us to a one-for-all type of therapeutic that we're pretty excited about, not only to help out the rare disease folks, but also a lot more people in general. So our lab works with adeno-associated virus vectors or viral vectors, and these vectors are very simple and comprised of a protein capsid and then just a single strand piece of DNA. Almost all of that DNA is actually transgenic or non-viral DNA. However, the ends, or the inverted terminal repeats, are about 300 nucleotides of viral DNA, which is the only persistent viral element of AAV vectors following administration. And we can think of this simply as a FedEx truck, where you put a gene that may help something in there and see what happens. Right, so to reemphasize, there are no viral genes to AAV vectors, and in general, they're not competent for replication. They have good safety profiles and durability following a single dose for the treatment of many different diseases, and there are currently 7 FDA-approved drugs, and this is, just simply since 2018. Our lab works with corneal diseases in part, and about 5 or 10 years ago when I got into ocular gene therapy using AAV, there wasn't really many groups in the anterior eye. So we set up camp, and it was really inviting for us. And I'll go ahead and put on the record that the cornea is perhaps the best tissue for AAV gene therapy. And there's a lot of obvious reasons for this. One, it's an external organ, so it's easy to treat, it's easy to monitor, and it actually can be removed. The eye in general has this weird phenomenon that we'll talk about further called immune privilege. And so the cornea, like the posterior eye, is actually an immune privileged tissue. It's easily transduced at low titers by multiple AAV serotypes. So, perhaps some work better than others, but all of them work fairly well that we've tested so far. When administered to the tight central cornea or stromal portion of the cornea, the particles are primarily restricted there, depending on the disease state, but in general, the particles stay in the cornea and they transduce what are called keratocytes within the stroma. There's a lot of collagen in there and not too many cells. And the stroma cells, or the keratocytes, are non-dividing cells, making it an ideal target for AAV gene therapy for long-term applications. Another cool thing is, we can do a lot of experimentation on human corneas. And, these are part of what is donated if you're an organ donor. And so we don't have any kind of species translational issues. And perhaps most interesting is that the conjunctiva, the cornea, is a natural reservoir for AAV in nature. So it's a very natural place that these particles actually infect in a natural setting. So the beginning of this talk, this is the all-for-one approach, was initiated by a clinician at Duke, Dr. Joanne Kurtzberg. She works with lysosomal storage diseases and performs hematopoietic stem cell transplantations as a treatment for those types of diseases. She crossed the aisle and came over to UNC and asked us probably 5-10 years ago if she could work with us to help develop a treatment using viral gene therapy for her lysosomal storage disease patients, in particular mucopolysaccharidosis type 1 or MPS 1. These children and patients in general have a progressive corneal clouding that results in vision loss over time. And this vision loss, especially in this population, really reduces the quality of life. And so the idea was this is something that her stem cell transplantation did not correct, and perhaps we would be able to do a different approach to help restore vision. And the gene causing MPS 1, it's a monogenetic disease, the mutations in IDUA. And then IDUA, to put it simply, encodes for a secreted enzyme that cuts large sugar molecules or glycosaminoglycans. And thinking about it in one way, the enzyme normally cuts down between a certain sugar linkage, and then it processes them into smaller pieces that can fit in the garbage can of the cell. However, if you don't have the enzyme, then you don't have the ability to cut those sugars, and you result in a storage disease. And so this storage disease actually results in extended or distended cornea, and this distension causes a loss of vision and progressive corneal cloudiness. There's no treatment for this. Here again, we have the blue representing the accumulated glycosaminoglycans in the cornea, and we see that there is actually increased GAGs and some degree of disorder in alignment. So we know this is a disease of the central cornea. And if we get closer, we can even see that in more detail. So the target is actually the central cornea. And the idea is that we can hypothesize that a simple IDUA gene addition strategy will prevent and reverse the corneal clouding. And this was what I call a simple gene addiction strategy. I mean, this, genes broke, put it back type of thing. And sometimes I've seen these work better than others. I won't go into this too much, but we did capsid surveys in human corneas And we kind of demonstrate that most serotypes themselves, natural serotypes, do well for transduction. And so in some ways, 8 or 9 or chimeric of 8 or 9 mediates really efficient transduction to the cornea at extremely low doses. So we knew we were able to get it in there. Simply made a expression cassette with IDUA expression. The pink shows normal IDUA within a human cornea. And then after a single corneal injection is only 6 or 7 days, you can see that we can certainly encode the protein via its cDNA within the vector. And we wanted to test whether this was going to work. And, like humans, dogs also get MPS I disease, and their phenotype is somewhat reflective of the human condition. And they do get the progressive corneal clouding observed in humans, although they have a lot more vascularization and other issues, but they do get the progressive corneal cloudiness. And so we asked in this naturally occurring model and worked with Keiko Miyadera at UPenn, a veterinary ophthalmologist, and our first round was in just 4 subjects. 3 had advanced disease, so significant opacity already. 1 was mild disease, and we did contralateral injections, 1 encoding the AAV IDUA, and then the contralateral including a fluorescent reporter. And we used a dose of 6 to the 10th in these first experiments. Here we can see that we have the mild opacity in the younger animal and the severe opacity in the older animals. And we did a single intracorneal injection, which is an administration route for fungicides and other things that are given or administered to the cornea. So fairly standard injection. And what we were able to see is that post-injection, within actually 2 weeks, there was 2 to 3 weeks, there was near complete reversal of the Stoller's disease. It didn't matter whether it was more progressed disease or not. The reversal occurred in all animals, independent of how severe the disease was. And so really kind of amazing result. And when we look at this, here you can see when I was treated and when I wasn't. We can see, the back of the eye very well and in very detailed because the cornea is clear. And you can see this, and this is reflected by the flash photography reflecting off the back of the retina. However, the same animal in the other eye that was untreated or treated with the control vector continued its corneal cloudiness to opacity. And so, we really were kind of blown away by these data, and then kept working this angle, and we did some more subjects. And when we did a dose de-escalation study, where we went over 2 log orders of magnitude for vector genome dose, and then we were able to see the clearance kinetics right there. So for instance, 1e to the 9th took 3 weeks to become clear, 1e to the 10th took 2 weeks, and then 1e to the 11th demonstrated near complete disease reversal within 1 week of treatment. So these are remarkable time points and remarkable low doses, and this dose-dependent effect is quite dramatic. Here we can see 2 weeks post-injection with a dose of 1e to the 10th that the cornea has already become clear compared to the contralateral cornea, depicted there on the right. So we've seen storage clearance in all treated MPS I eyes to date, and the clearance kinetics correlated with dose. And we have 10 subjects that have been treated successfully with little to no adverse reactions over this for the efficacy of this program. To keep in mind, a dose of 1 to the 9th is extremely low. Luxturna is an AAV-based gene drug for a rare form of retinal degeneration. These vectors are administered by the subretinal route of administration. And in those cases, for a little pocket behind the retina, they use 100-fold more virus particles than we're using in the cornea to get a quite profound reversal of the disease. And then theoretically, one prep can treat all MPS patients now and into the hundreds of years of the future. And so, at this point, we had discovery, we had efficacy, and with some safety profile in a MPS large animal model. And then we did a rigorous safety studies and extreme biodistribution, neutralizing antibody to the capsid induction, enzymes and all that. Everything looked good. We actually went to the FDA. We got positive pre-IND feedback. And so everything was going really cool. And then, and just put this in perspective, this took about 6 years up to this point. Several publications, a lot of money and costs, but we worked well and cheap because it wasn't funded that well through the NIH actually at all. And so we worked with the Society of MPS Society and other folks to get enough money to do these important studies. And as we were going, we got positive pre-ID feedback, everything's looking good, and then the fruit just sat on the tree and rotted on the vine, and years start going by, and the problem here is, at least at that point, there was low commercial interest for rare disease gene drugs, and so we needed millions of dollars to not only have the GMP prep produced, but also to be able to run the trial at a hospital and pay all the folks that needed to be paid. And despite this, eye-popping data, sorry guys, we weren't able to get significant investors or commercial interest to move this forward. So talk about frustrating, and feel like you kind of let folks down because it was so promising. So we had to find a way to get around this and to try to stay with our focused objective to help MPS I patients. And so, but this low commercial interest for rare disease gene drugs is not that ridiculous. I'm calling it the reality roadblock, that now that I look back years down the road, I probably should have thought about not going into the cave without knowing there was an exit on the other side. But thinking about this, all right, so each disease is going to need its own therapy. And for these rare diseases in particular, there's low investor interest. I put 5 to 20 million for the development and to run a trial and get a GMP prep. Those numbers can vary, but that's probably standard. And, at the end of the day, there's probably a low return on investments for any person or company that put in the money to actually develop it. So, and then furthermore, we did this in maybe 5, 6, 7, 8 years. We've done it again for another MPS disease in a large animal model. It took us 2 or 3 years. So the timing is variable as well. But, that investor risk is if it works, and so, and then there's no adverse toxicological effects. And so there's a gamble with just the efficacy, with the safety, and then, trying to recoup the money that you put into it. So in some ways, it can be rationalized why folks don't want to invest much money in these types of approaches. And then just doing a numbers game a little bit here, there's 8 other lysosomal storage diseases that impact the cornea, in which there's progressive corneal clouding until vision loss. And so that means we'd have to have 8 more drugs in addition to AAV IDUA just for the lysosomal storage diseases. And, you could do the numbers in your head and how much effort this is and for what's the return. And it's getting a little bit, daunting in some ways. And so if we look at all the rare diseases out there, I mean, there's 7,000, there's 250 new described annually. There's well over, probably 7 to 10,000. And then, and with 50% of these affecting children. So when contemplating a gene addition strategy, thinking that we're going to have, especially for rare diseases where there's not much commercial interest, but we're going to have to make an individual reagent for each one of these diseases, assuming they're monogenetic, so we can cut that number in half. And I'm not sure it's making sense. And so here we have some thousands of new AAV drugs, and so approximately 7,000 diseases. One could say if they're all monogenetic, maybe 7,000 new drugs. This is hundreds and hundreds of billions of dollars for AAV gene addition drugs for rare diseases, when there's not much commercial interest, even if you have stellar data in large animal models and pre-IND feedback. And so, this is, in some ways, I don't know if this is a practical approach. And so at least not in my head and what I've experienced so far. So we have to think about a way to get around this and come up with an alternative strategy. And we didn't know this going into it, but our trajectory here was to target some low-level conserved or shared etiology, and then try to address that, and hopefully it would take care of some other problems. And in the case of MPS I corneal disease, these patients are able to undergo corneal transplantation, but they're often rejected, and it's really reserved to the last case or the last hope. And that's highlighted by the fact that this was a clinician-inspired project where that clinician is well aware of the high risk of rejection in MPS I subjects. But they do have that possibility. And so here you can see that corneal transplantation is very common, actually. It's the most transplanted organ besides blood, and worldwide. And there's 50,000 performed in the U.S. annually. Greater than 60% of these transplants are high risk, because why would you do a low risk transplantation? Because you may be able to keep that in the eye longer until it got high risk. So there's a lot of these transplants are high risk, and the MPS I patients are considered that high risk as well. And the high risk of rejection is evident with the numbers vary crazy, but 30 to 80% at 5 years of high risk corneal transplants are rejected. And so thinking about the MPS I patients, we went ahead and tried to revise our objective, and instead of treating them with a bespoke form of gene therapy that nobody was willing to invest in at that point, maybe we can just prevent corneal transplant rejection via this idea of immune tolerance. And so if you look at the corneal transplant rejection, these are common figures. Again, most of them are rejected. There's a lot of folks on the waiting list for tissues, and there's about 50, and a lot of those people don't have access to tissues. There's no banking or anything. So there's a couple problems, and one is the high rate of rejection. The other one is the tissue availability. But thinking of the idea of immune tolerance, and how can we make a foreign tissue be tolerated in another person that's genetically dissimilar, we came up with the idea of perhaps the only natural, or one of the natural instances of allogeneic coexistence is pregnancy. And here's an interesting sketch from Nilsson et al. And what they demonstrate here is that the baby developing in the mother is protected by a protein that is highly elevated in pregnant women. And this is the HLA-G protein. The HLA-G protein protects the baby. It's all over the placenta. It's all in the mother's blood. It's everywhere. So the mother is actually immune suppressed by HLA-G in part, and the fetus is protected by HLA-G in part. So, and this isn't only seen in a developing fetus, it's actually seen in seminal fluid and pretty much any time that you would have foreign cells going into a different genetically diverse person, and then you would want to protect it. So HLA-G prevents fetal rejection via the induction of immune tolerance. And so, at first this was actually brought to me by a friend and collaborator, Cheng-Lin Lee, at University of North Carolina. And he's a brilliant immunologist. And so this whole idea of, protecting the cornea, like the body protects the fetus, seemed to make sense. And then, as I got into the ocular space, there's a lot of talk of ocular immune privilege, ocular immune tolerance. And this is kind of a natural state of the eye to suppress inflammation and to suppress vascularization. And that by doing that, it blocks the immune system from coming in and causing problems. Any inflammation in the eye can result in loss of vision. So you want to suppress the immune system in the eye. And here was a crazy study interesting study, from 1948 by Dr. Medawar. He took pieces of skin and, allogeneic skin, and put it in the brain of a rabbit, put it in the anterior chamber of the eye, a piece of skin, and then stuck it in underneath the skin of a rabbit skin, and then just looked to see what happened. And there was actually no rejection to the allograft within the eye. And this, in part, started this whole concept of ocular immune privilege or ocular immune tolerance. And then later on, by Dr. Carosella is kind of the godfather of HLA-G research and understanding its functions. Decades ago, that group published that HLA-G is in the human cornea, and then connected it with this idea of ocular immune privileged tissue. And it's like, wow, things are just seemingly to come together. And so thus far, it seems that HLA-G is involved in fetal immune tolerance, protecting the baby, and then ocular immune tolerance, protecting the eye from inflammation and preserving vision. So super interesting. But if you look at some HLA-G functions, it's probably one of the most potent immune modulators in the body. Actually, it works directly and indirectly on all aspects of the immune system. The molecule itself can bind receptors on the depicted cells, almost all immune cells, to inhibit them directly or induce apoptosis, thereby inhibiting the immune response. And there's membrane isoforms, there's soluble isoforms, and then the soluble isoform is thought to go away from the cell, and it can actually prevent neovascularization. It can also degrade pre-existing vessels. And by doing this, it's a secondary or indirect measure or manner to inhibit the immune response. And so as HLA-G in human corneas, we had a source of human corneas. So we went ahead and did some Western blots. The first group of corneas, it seemed that everybody has it, but maybe at different levels. The next group of corneas, despite having our positive control there, we weren't able to see any HLA-G is in this set. And then we did a couple more corneas by Western blot, and then we saw different levels of HLA-G. So HLA-G positive corneas at this point were about 70% of the corneas we sampled had HLA-G in there. So similar to Dr. Carosella's paper and report, we did see differential levels in the human cornea. We tried to correlate it with age, gender, sex, things like that. but it's not so clear yet. But it's maybe something to do with increased HLA-G in the cornea over time. And it's unknown. But we're like, okay, well, it's there already. Maybe we can do like a complementation or an augmentation strategy to deliver HLA-G and then use it for different ocular diseases. And so that's what we did. And this was in part with my these experiments were performed along with Brian Gilger, a long-term collaborator at NC State. He's a veterinary ophthalmologist and toxicologist, and has experience with a lot of animal models of ocular diseases. And we demonstrated that AV-HLA-G in various reports does a lot of different things. It can be applicable to a lot of different ocular diseases. I mean, trauma-induced corneal neovascularization or fibrosis, AV-HLA-G was able to inhibit that in the vascularization caused by trauma. Ocular graft versus host disease, kind of a rarer type of disease following stem cell transplantation, but a good, somewhat of a model for a dry eye disease, which is more common. And AV-HLA-G was able to inhibit that as well. And furthermore, autoimmune uveitis, it's one of the leading causes of blindness. And then treatment of AV-HLA-G proved effective at decreasing the severity of symptoms associated with that. So just by targeting just anti-inflammation and anti-vascularization, we're able to apply the same drug to different disease models of the ocular diseases, and it seems that it's effective in several ways. As we kind of kept reading and doing the literature searches and stuff, it was clear that the optimal function of HLA-G is elicited as a dimer molecule. And this dimer can be a homodimer, in which it binds itself, or a heterodimer. There's many different isoforms of HLA-G, but here we have an isoform that mimics the soluble HLA-G5 monomer with the alpha 1, 2, and 3 domains. They come together, they have function by themselves, but for optimal function, increased receptor affinity, they actually dimerize through disulfide bond formation at this position here at the cysteines. And then this is the active molecule. And folks have been trying to produce a single polypeptide that encodes the HLA-G homodimer for a while, because this step is deemed rate limiting as far as HLA-G function. And so our idea was then to design a single protein to mimic the HLA-G homodimer, thereby overcoming this rate limiting step. Here we have a secondary protein structure of the HLA-G homodimer, again the disulfide bridge in the middle, and I have in a purple square an HLA-G monomer. Working with Brian Sutton, a long-term collaborator and crystallographer and everything about protein type of guy, he was able to generate a polypeptide sequence that, at least through structural prediction, appears to form an HLA-like molecule. And to do this, he kept the alpha 1, 2, and 3 domains the same, and he connected the alpha 2 and alpha 3 of alpha 1 and alpha 2 of the other dimer molecule by having this link or kind of a spacer or conformational type of protein amino acid sequence derived from a Providence virus coat protein. And this is an IG-like domain. And so we went ahead and, cloned this molecule, synthesized this molecule. Here we have another picture here of it. The linkers he added some adapter sequences as well. And the structural model kind of looks like the HLA-G homodimer. And so we engineered this, we did some Western blotting. It indeed is a stable protein. We can see it there about the predicted size. And then we were also able to see it secreted in the supernate. And so this was a secreted molecule that's supposed to be mimicking the HLA-G5 homodimer. And we didn't know if this had any function, so we had to go in there and see if this would work. We did a corneal trauma model And what we could see by the vascularization score is that there's decreased vascularization in the AAV skim treated eye. And you can see here quite dramatically, almost minimal to no vessels following corneal trauma in the mice receiving a subconjunctival injection of AAV skim. And the histology there, and I apologize if my head's right in the way of that picture, but the treated corneas or treated animals actually showed very little to no immune cell infiltration or signs of vascularization, in contrast to the vehicle, which was traumatized. And so the whole point of this was actually to go after, the corneal transplantation, high rates of rejection, because our objective actually was the MPS patient population. And so now that we're thinking about this, we're going to try to prevent high-risk transplant rejection via the installation of immune tolerance. I'm using this single-chain immunomodulator, or SCIM protein, that mimics the HLA-G5 homodimer. And the cool thing about this, in some ways, at least from my perspective, is that we can do an ex vivo gene therapy approach. So we're not treating or dosing patients. It's simply tissue engineering, and the idea, the strategy, is to treat the healthy graft, you wash it off before we put it in, and then put it into a high-risk model, and then let's see what happens. And that's going to decrease increase the safety profile, decrease the biodistribution outside of the eye, and really kind of making it a more feasible type of approach. To put it in a different depiction there, we have a healthy corneal graft. We incubate or marinate it with the AAV skim molecule, or particles, encoded in the AAV particles. We wash it a few times in saline solution, and what we're able to see there, and I apologize again if my head's blocking it, but we do get particles bound to the tissue, but it's at the order of 1 or 2 e to the 6 vector genomes. So this is a very, very, very low amount. This is 4 logs lower, or 3 logs lower than we're using in the large animal corneas. And so while, not too much virus, we hypothesized this could be a potent molecule. So we went ahead and did it anyways, even with that low dose. And so 15-minute incubation, we created a high-risk model. We let that become high risk over 14 days by putting sutures actually into the cornea to generate a situation that's similar to the human condition. We took the healthy graft ex vivo incubation with AAV skim for 15 minutes. I'm getting about 2 e to the 6 particles attached to the tissue. We wash it off and we do the transplantation. Extensive analyses. This work was recently published earlier this year, and I think it's why we got the invite to present in this forum. And what we were able to see is that after the 17 days after the transplantation, this is an inflammatory score by a blinded clinical ophthalmologist who examined these animals. There was no change in the treatment group or PBS, but after day 17, the inflammation in the corneas that received AAV skim went essentially to baseline. These were quiet and healthy looking clear eyes, and then in contrast to the inflammatory score in those that were just received the saline. Corneal thickness by optical coherence tomography is actually a completely objective way to measure things. It measures central corneal thickness. You can see that the PBS or the skim treated corneal grafts behave the same, but then the central corneal thickness went down to baseline in skim, and compared to the one in the PBS, which actually just kept getting thicker to about 3 or 4 times what a normal cornea should be. The graft survival rate was near 100%. It was 100% for every every corneal graft that was treated with AAV skim was well tolerated, and in contrast to those that received saline, all but one rejected. And I hate that that's the case, but I encourage you, if you're interested, to look at the PMID number there. You can see the vehicle central cornea is opaque, cloudy, and perhaps around my icon in the bubble, you can see that the AAV skim treated cornea is not, like I said, it's not rejected, it's not inflamed, it's clear, and it's normal thickness. If you look at the actual histology, a vehicle, you can see thickness, you can see areas of severe infiltration, whereas the AAV skim cornea looks normal. We have the incision site in the central cornea there on top with the vehicle. You can see that there's a whole bunch of immune cell infiltration. There's vascularization, vessel cross sections we can see in there, whereas the central cornea and the skim treated graft looks perfect, actually. At the incision site, we can see that it actually was transplanted. So there's evidence that this wasn't just a healthy cornea, because you can see the scar there. But overall, it looks quite clean. And interesting too, we saw a decrease in overall fibrosis, which we did not see much at all to any fibrosis in AAV skim treated corneas, and that's compared to the vehicle. Biodistribution, it stayed in the eye that the treated graft was implanted into. There was no induction of AAV antibodies, nor would we expect them to be with 1 to the 6 vector genomes associated with that corneal graft. So overall, skim is a stable protein. It prevents trauma-induced corneal neovascularization and also high-risk corneal transplantation. And here we are with our progression towards, our original objective, which is the MPS 1 patients. And additionally, below the MPS 1 patients, it says in thousands and thousands of more, because this is a very common procedure. And we're hoping to get to this type of treatment in the clinic soon. So I think for me, kind of the surprise part was, we banged our head against the rock literally for 2-3 years by taking an alternative path to help our focused objective of MPS 1 patient corneas. We were able then to come up with a treatment that's at least one for many. And that includes the actual MPS patient population. And so to date, AAV-HLA-G or SKIM inhibits several diseases as discussed. With that, I'd like to thank my collaborators Chang-Wen Lee, Brian Sutton, Brian Kilger, my boss, or my chair, Don Budence, Keiko Miyadare, who did the dog work at UPenn, Jude Samolsky, and Joanne Kurtzberg, who were there to urge this project forward on the early side. Also the lab and those who have funded it or supported it over this time. And with that, I'd like to say thank you again. And you guys can always feel free, if there's interest, to write me by email. I'm happy to answer questions in this form or a different one. So thank you, and I'll take care.
Perfect. Thank you so much, Matt. Let's jump into the questions. Will extended ocular immune suppression result in a greater propensity for ocular infections?
Yeah, that's a great question, and it's one that we haven't thoroughly tested. There's a possibility, I think, at least for the corneal transplant, that it's in the environmentally exposed tissue, but I think the treated cells were actually the ones in the central cornea, and the epithelial cells of the cornea actually die over time in pretty much a short lifespan of like 5 or 6 days. So I don't think we're in that case suppressing the corneal surface. So I don't think there'd be a major threat of infection. As far as the central eye for like uveitis or something, it remains to be seen. But the current treatment is steroids, and steroids actually result in increased infections as well. So hopefully the AAV treatment herein would immunosuppress the corneas and the internal eye without causing too much of an increase in infection, and also not have the adverse reactions that are associated with commonly used steroids.
Excellent. Thank you. For the gene addition strategy to treat MPS corneal blindness, what was the duration of the clearance of storage disease?
Yeah, because of the nature of the model, which has other systemic disease manifestations, the humane endpoint was about a year and a half to 2 years old. So we took it out a lot of those studies about a year. One of the things that I think when we inject all those particles in the cornea, I think a lot of them actually don't infect cells, and the intact particles just hang out there with the ability to maybe infect at a later time. So it's almost like a reservoir pocket. So while we don't have any data beyond a year, I would suggest that we would expect it to last much longer.
I see. Thank you. Do pre-existing neutralizing antibodies to the AAV capsid prevent corneal gene therapy?
Yeah, really common question, and we don't have conclusive evidence for this. But in another model of lysosomal storage disease, MPS 6 felines actually, we did sequential dosing of the first cornea. We waited 4 to 6 weeks, administered to the second cornea, and we didn't see any decrease in the therapeutic effect or the clearance kinetics. I think it maybe depends on the disease state. If there's vascularization and there's blood and there's potential for antibodies in the cornea, it's a possibility, but I think it's a low possibility.
Excellent. Can the skim molecule that you're discussing in the second half of your presentation, do you think it can prevent or might one day help prevent heart, liver, or kidney organ transplant rejection?
The hope is yes. We don't know, and at this point, we started pretty much at the easiest organ that is rejected in the cornea, and we are working in discussion with folks at Duke and to do look at maybe heart transplant rejection, and then maybe walk down the different organs over time. So I think the possibility is there. It's interesting because for heart transplantation and for liver transplantation, the likelihood of not rejecting the organ correlates to the level of HLA-G in your blood, such that if you have high levels of HLA-G in your blood, then you are more likely to tolerate foreign organs. So perhaps from just those observational studies, it's suggestive that it could also be applied to other organs.
Fantastic. And maybe time for one last one. Could you tell us a little bit about the future avenues for your work that excite you the most? What are you looking forward to?
Yeah, related to this project and the data presented herein, I'm excited about a lot of other things as well. I think the corneal transplant, if we think about transplantation in general, there's two major things. It's kind of the organ availability and the high rates of rejection. So in some ways, I think the future of this work, as others are doing, is to think about transgenic pigs and the idea is, transplantation and thereby taking care of both the tissue availability and the potential for rejection of a foreign tissue.
Perfect. Thank you so much, Matt, for that wonderful presentation and also that great discussion.
Thank you.
And now it is my pleasure to introduce our final speaker. Thomas Quinn received his Bachelor of Science degree in zoology with a concentration in genetics from Michigan State University. He completed his Master's of Science in Molecular Medicine and Genetics from Wayne State University, where his thesis focused on the development of viral vector systems for application in CAR T therapy directed at melanoma and lung malignancies. Since 1998, he's been working at Takara Bio USA as an R&D group leader, focusing on the development of high-efficiency viral delivery products including adenoviral, retroviral, lentiviral, and AAV systems, as well as CRISPR-Cas9-based genome editing products. Let's make sure that our slides are up and running. Excellent. Take it away, Thomas.
Hello, my name is Thomas Quinn. I'm an R&D group leader here at Takara Bio USA. Thanks for joining today. Title of my talk is Redefining Tools for AAV Applications. First, I'd like to start by introducing a little bit about Takara Bio's history in advancing cell and gene therapy with high-quality viral delivery tools. It started in 1995 with the introduction of retronectin reagent for the enhancement of retroviral transduction. It was followed in 1998 with the introduction of the product line RetroX that focused on retroviral vectors as delivery tools. And it was our first comprehensive offering. It was followed up in 2003 with our AdenoX product line, then in 2008 with LentiX, and 2013 with AAV Pro. We've continually developed these gene delivery tools as exemplified by our introduction in 2018 of the LentiX Go Sticks, which is a lateral flow titration tool, and I'll talk a bit more about that in a moment as it applies to AAV. And then we also most recently introduced the LentiX transduction sponge, which is a dissolvable cake to simplify lentiviral transduction. That was introduced in 2024. Lastly, we also had an expansion of our GMP and CDMO viral vector manufacturing capabilities in 2020. Throughout all of these product lines is a high maintenance of quality because we follow the ISO 13485 standard. And as I forgot to mention, we're headquartered in San Jose, California. But the focus of the talk today is our AAV Pro tools, including AAV vectors, titration, and packaging technologies. So just to cover a little bit of the background here, I think we're all familiar with this, but I'll run through this briefly. AAV has become a really popular vector of choice recently. It's a single-stranded DNA genome, approximately 4.7 kb in length. It has low immunogenicity. It's non-pathogenic. It's relatively small compared to lenti and adenovirus at 25 nanometers. The capsids, icosahedral in shape, product of the VP1, 2, and 3 proteins. And it specifies the serotype. And the serotype is important because it determines the tropism or the types of cells that it can infect. And each serotype exhibits a different tissue specificity. And of course, the efficiencies are based upon that. The applications obviously are quite broad, including gene function studies, gene therapy development, disease modeling, and CRISPR applications. Now, if you're looking at a mammalian production system for this vector, it usually consists of 3 vectors: a transfer vector, which is containing your ITRs and your sequence of interest; a rep-cap vector, or replication and capsid vector. This expresses the main parts of your viral particle. And there's also a p-helper vector here that helps the expression of these products within the transfected cell. These 3 vectors are then transfected into 293 T cell, where the vectors are then transcribed and translated to produce both the genome and the particles. At that point, the genome gets packaged within those particles, and after a while, the cells are harvested, and the soup is harvested to collect those particles. Depending on which serotype it is, of course, determines if the virus is in the soup or the cells. Speaking of serotypes, I have a figure up here on the right, just looking at the breakdown of the different serotypes, looking at AAV9, AAV8, and AAV2. And together these make up 60% of the serotypes in use, and it's mainly because of their tropism. AAV9 has applications in the brain, across the blood-brain barrier. AAV8 is good for liver delivery. AAV2 is used for CNS and other applications. So depending on the type of serotype or tissue you're trying to target will determine what serotype you're going to use. And one thing I failed to mention here was this 18% of others. A lot of work is going on into modifying these wild type serotypes to make these vectors very specific for the tissues that are being targeted. ablating those targets that are not wanting to be transduced, and targeting more effectively towards those desired targets. Now, producing these is a multi-step process. We have products for each of these, including starting at the vectors. We have those that have CMV constitutive promoters, TET-inducible, also those that can be used for editing, such as Cas9 or Cre recombinase. And lastly, we have reporter vectors that can be used for optimizing vector production and purification. For cloning into those vectors, we have the infusion SNAP assembly master mix with competent cells. That's a ligation-free cloning, PCR-based cloning product. And then once that vector is completed, that needs to be packaged. We have a number of different packaging systems for those, including the AAV Pro helper-free systems for serotypes AAV1, 2, 5, and 6. Those will be packaged in the AAV Pro 293T cell line. And what I'll talk a bit more about today are the packaging single shots and the backpack GhostX Plus. Once the particles are produced, they need to be extracted, concentrated, purified, and we have a number of different purification kits that also concentrate. I'm not going to go into too much detail on these today, but once that product is concentrated, purified, it then needs to be titrated, and we have a number of titration kits, including one real-time PCR kit, the AAV GhostX Plus for 2, 8, and 9 serotypes, as well as the AAV ELISA Rapid Titer Kit single wash for AAV 2, 8, and 9. And so, as I mentioned, these will be the ones I'll be speaking about today. And so the reason we chose to work or present these today is we want to simplify this process for those that aren't necessarily as experienced in using the AAV as a viral delivery tool. And so we're doing this through an introduction of a simplified packaging solution, the AAV packaging single shots. It's a one-step packaging mix. The AAV GhostX Plus, which is a 10-minute titration product, and then the AAV ELISA Rapid Titer Kit single wash for fast and accurate titration, especially when you're doing higher throughput vector titrations. And so combining all of these leads to a much simplified workflow. So starting out with the AAV packaging single shots, what do these consist of? These are basically a single tube that contains the packaging constructs for the rep cap and helper vector, along with the transfection reagent exfect. These are then lyophilized into a mix at the bottom of this tube, so that you simply have to add your transfer vector and water to create the transfection mix that then gets applied to the 293 T cells for transfection. This is simply a 10-minute incubation after addition of your vector. This is in 3 mils volume. And the number of cells that this will transfect is 2 P150 tissue culture plates. This transfection is then incubated for 4 hours to overnight at 37. Additional media is then added, and the viral particles are harvested 72 hours post-transfection. And to give you an idea of the performance of these transfections, we have packaged a ZS green expressing AAV vector using serotypes 2, 8, and 9. And this is comparing the transfection efficiency of this lyophilized transfection mix to that of a liquid version of AAV2. And you can see it's comparable across all the serotypes to that liquid formulation, and that they're all above 95% transfection efficiency. This is measuring fluorescence at 72 hours by FACS. Now, when those products are harvested, we then collected the crude lysates from these, as well as the supernatants, and we then determined the crude vector genome yield using a qPCR kit that we market. And in each of these cases, you're seeing we're getting greater than 9 times 10 to the 12th vector genomes per mil of the crude virus product at that point. This is what you would be putting into your concentration and purification protocols. So the yield is very good off of all these. It's very consistent. So once you've produced and isolated your particles, you're then going to want to titrate them. And this is going into the AAV GhostX Plus. Now, the reasons for titration are pretty important, actually. The first being that it verifies your particle production. You need to make sure that there's a lot of moving parts here, and you want to make sure that you're getting a significant yield from your transfections. Secondly, it's ensuring experimental consistency. This allows you to dose your transductions at consistent levels, so you can control the MOI in each of the cases, leading to consistency from experiment to experiment. It also allows you to improve your purification method, so you can now harvest your flow through, your wash, your elution, and see where you may be running into trouble in terms of yield, which will lead to higher yields when the process is improved. And lastly, you can look at product quality. By doing titration assays, you can assess the empty to full particle ratio to look at the potency of your AAV prep. Now, there's a number of different titration methods here, going from the longest to the shortest. And at the top here is your infectious unit assay, where you're looking at transduced cells, and that's if you have a marker which you can actually assess in the transduced cells. There's also analytical ultracentrifugation here, electron microscopy, blotting, ELISA, dPCR, qPCR, and of course the last one, the Go sticks, which I'll talk about in a moment. But each of these methods has their workload, and the ELISA, the qPCR, and the Go sticks are methods that we actually have products for. But looking into the Go sticks, how do these work? Essentially, you're taking 20 microliters of your prep, whether it be crude or purified. You're adding that to the stick. You're then chasing it with 80 microliters of chase buffer and incubating for 10 minutes. What's happening during those 10 minutes? Your sample is then migrating through the membrane to a point where there's actually antibody that's conjugated to gold particles. These complexes then migrate further down the line to a second antibody, which then captures those complexes and deposits them at that test line. If there's any unbound gold conjugate, this migrates up to the anti-IG control line here and is deposited there. That's why you have the two bands. And so what that looks like here is you see your positive result here, you have your negative here. This is then quantified using a smartphone device, either Android or iOS. And that app is free in both stores. This app then quantifies, it measures the intensities of each the test and the control line, performs densitometry on those, and you can see that here. This ratio of T over C is then used to compare to compared to a lot specific standard curve, and that turns out the Go stick value, which in this case is viral particles per mil. And this can be compared to other titration methods such as qPCR and ELISA. And so this is a data set that was produced using an AAV2 vector expressing ZS green. And we did just that. We took the prep and we titered it by 3 different methods. We measured the infectious unit titer, we measured the qPCR titer, as well as the Go sticks titer. And this was done for 3 different lysates, as well as a purified vector. And the reason I included the purified here was just to demonstrate that when you use an antigenic assay, such as an ELISA or a Go stick, whenever you do purification versus the crude, you're actually changing the antigen levels within those preps. And so when you start comparing them, you actually see a reduction in the antigen level, which you'd expect when it's purified. And that can change the relative ratio between these. So as we move forward here, it's important that when you do these comparisons, and I'm going to show you, that they're all comparing preps that were made using the same protocol. That includes whether it's crude or if it's purified. So in this case, we took lysate 3, for example, and we compared the Go sticks readout to the qPCR. These values can then be made into a relative ratio between these. And in cases where, say, for example, lysate 2 and lysate 1, you only had the Go stick value, you can multiply it by this ratio to get a calculated qPCR value for these 2 preps, especially in the case where you're used to using vector genomes per mil to determine your MOI. So you can continue using that value, however, it can be calculated off of a Go stick. And I'll show you here, if you do this calculation, here we did it to the right, the lysate 3, which is used as a reference here, is set at 1. But if you do that calculation, the calculated value turns out to be less than twofold different from the actual value here. So what I'm saying is that you can actually use this 10-minute lateral flow test as a surrogate for your qPCR readout and still have confidence that your MOIs are going to be accurate. We took this one step further and looked at serotypes 8 and 9, again using a ZS green expressing vector. And again, the value calculated from the GO stick was very close to that of the actual ELISA value across 3 different samples when using the 4th as a reference. And we only use the 4th here. Most labs have a marker virus or a virus that they're used to using on a regular basis. They have the vector titer by some means, whether it be qPCR or ELISA. In this case, these vectors were all produced the same way, and we use that as a reference. But it was similar for 8 as it was for 9. These ratios were all very, very close to what we saw for the AV2 as well. One thing I failed to mention before was that majority of people are using the mammalian system for AAV production. However, there is a growing contingency that's using baculovirus because of its scalability and consistency. And in those systems, they're basically using the baculovirus to deliver the cap, the rep, and the ITR containing vector. And there's different designs for these where the rep cap are the same virus, or all 3 are the same virus, but this is just a schematic to give you an idea of what we're talking about. Now, when you have these virus particles here, the baculovirus, these also need to be quantified, and we also have a lateral flow test for that as well. And this is what we did here, where we tested a number of different clones that we had isolated, where we did plaque isolation, and then we titered these using the lateral flow test. And this is just showing you the T over C ratio for each of these based on the signals. And just like for the AV8, 9, and AV2, we have a calculated value for these clones compared to the plaque forming units that were actually observed using the rapid titer kit, where you stain plaques and count plaques. So essentially, we've taken a 2-day assay and shrunk it to a 10-minute assay when you're looking at baculotiter. But again, you can see how robust the workflow is, because it applies to baculo as well as AAV, and of course to lenti as well. Now, in cases where you have a lot of samples to titer, or you have multiple replicates that you want to run, the ELISA is a very good solution for that. And for this, we have the AAV ELISA Rapid Titer Kit single wash. And in this particular product, we're able to eliminate a number of the steps and simplify the workflow while maintaining the robustness of the assay. So in this case, we're adding the AAV along with the detection antibody, placing that in the capture well, incubating this for 1 hour at room temp. We then wash, one wash, and then add the substrate, incubate for 15 minutes, and then add the stop, and then read. And the really nice thing about this is that it does reduce the steps and hands-on time. It still maintains the accuracy, which I'll show you next. It's compatible across multiple matrices, so you can titrate and cell lysates, elution buffers, and expression media. And it's automation friendly, obviously, because it's in a 96-well format. So you can titrate AV2, 8, 9 when you have liquid handling equipment. So just to give you a comparison of the titrations of this workflow, we compared our single wash kit to 2 other kits that utilize a bind wash, a multiple bind wash workflow, and you can see here that our data is maintaining the accuracy compared to the other 2 methods, but with a much shorter workflow. This is using an AAV2 GFP vector. And then just to look at the other serotypes here with 2, 8, and 9, and looking at the range of concentrations this can detect across, up to 4 times 10 to the 9th. Limit of quantitation for all these is 10 to the 7th. So have 2 logs of over 2 logs of a range that you can titrate across. And then when you compare the results of these different vectors to the qPCR here, you can see that they're very, very close to what you'd expect for qPCR. And in this particular case, this will allow you to calculate the empty to full ratio pretty quickly. And lastly, we've tested across a number of different matrices, including for the three different serotypes, 2, 8, and 9. We looked at the elution buffer from one of the purification kits, cell lysate, as well as media. And you can see we're able to maintain our percent recovery across all these different matrices that we've tested. So just to quickly summarize, we've streamlined the workflow for producing AAV by making the packaging a single step by introducing the AAV packaging single shots, which eliminates the need for packaging optimization with this lyophilized pre-aliquoted mix. Produces high titer and a one-step viral packaging reaction. Now, titration also is quickened by using the AAV GhostX Plus. It reduces hours of titration time to 10 minutes using a lateral flow-based technology to quantify those preparations. And if you have a increased number of samples, recommend the AAV ELISA Rapid Titer Kit single wash with improved workflow efficiency with the single wash, and it enables rapid and sensitive AAV titration in a much simpler workflow. So that's good science, and I appreciate your time. Thanks.
Excellent. Thank you so much, Thomas. Let's go ahead and jump into the Q&A. What factors can affect the readout on the GhostX?
No, that's a good question. Can you hear me okay?
Yes, I can hear you perfectly.
Well, as I mentioned before, the levels of anytime you do a treatment post-transfection on these AAV preps that alter the antigen levels, they're going to affect the readouts on the test. So it's just a matter of making sure that you're comparing apples to apples when you're doing your comparisons. So if you're doing an optimization of a purification protocol, you would compare the crudes together, the flow-throughs together, and the elutions together. So that way you're ensuring that you're seeing the same sort of matrix as well as the same sort of antigen levels. That way you can determine if the antigen level change is actually real.
Great, thank you. Will the GhostX work on serotypes other than 2, 8, and 9?
Yeah, I guess I failed to mention. So these are all individual tests. So one is for 2, there's another for 8, and another for 9. We are still in development with these, and while they work fantastic for those serotypes, we're expanding our range of these, looking at, say, PHP, DJ, and GATI, just trying to see if there's enough cross-reactivity to these tests to see if we can expand into those areas as well.
Great. Well, thank you so much, Thomas. Thank you for that presentation and for that discussion.
Thanks for your time. I appreciate it.
That concludes today's presentations. On behalf of the scientists, I'd like to thank our whole panel for sharing your work: Nerea Zabaleta, Hirotaka Kuroda, Matt Hirsch, and Thomas Quinn, and our symposium sponsors: Vector Biolabs, Shimadzu, Takara, Sinobiological, Beckman Coulter Life Sciences, Biorad, Taconic Biosciences, PHCBI, BPS Bioscience, AdGene, Innotiv, and Mission Bio. If anyone has further questions, please do consider reaching out to the speakers directly. Their contact is shown on the screen. As a reminder, the entire symposium will be archived on the scientist website, and you'll receive an email notifying you when the recording is available. Please also make sure to follow the scientist on social media for updates on future events. I'd like to thank everyone who took the time to join us today, particularly those of you who shared your questions and comments. And take good care. Thanks so much, everyone, and goodbye.